In performing manual hematocrit determination, fill 2 capillary tubes with blood and seal it using the sealing clay. Once the capillary tubes are sealed, place it in a microhematocrit centrifuge. Remember to balance the tubes inside the microhematocrit centrifuge and set the timer for 5 minutes. Also, do not forget the placement of the capillary tubes in the microhematocrit centrifuge. Once centrifugation is done, use a capillary microhematocrit reader in determining the patient's hematocrit level. The image shows the formula for the determination of the hematocrit level of the patient. For the hemoglobin determination, first fill the graduated tube with hydrochloric acid up to the 2 mark. Using a pipette, draw blood up to the 20th mark and expel to the graduated tube. After dispensing the blood, mix and allow it to stand for 10 minutes. Afterwards, add hydrochloric acid drop by drop and mix every after addition of the reagent until the color matches the standard or the color in the hemoglobinometer. Once the desired color is achieved, measure the hemoglobin of the patient by reading the result in which the lower meniscus touches the specific graduation on the tube. In performing WBC count, fill the WBC pipette with blood up to the 0.5 mark. Then, add hydrochloric acid up to the 11th mark of the WBC pipette. After aspirating the blood and hydrochloric acid, position the WBC pipette horizontally and remove the tubing. Also, do not forget to mix it. You may also opt to use a pipette shaker in mixing the contents of the WBC pipette. In dispensing the solution in the Neubauer slide, first place the cover slip on the Neubauer slide. Then discard the first 3 drops of the solution before charging it to the Neubauer slide. Remember to place the tip of the WBC pipette on the edge of the counting chambers and allow the mixture to be sipped and spread out on the chambers. Then let it stand for 1 minute before reading under the microscope. When performing microscopy for WBC count, remember to count on the 4 WBC squares of each chamber in the Neubauer slide. Also remember to count under the high power objective. The succeeding images show the appearance of the grids of the Neubauer slide in the portion where to count the WBCs. Shown in this image is the formula to be used in calculating the patient's WBC count. For the WBC differential count, use the battlement pattern in counting the white blood cells. Remember to count under oil immersion objective and count until you reach 100 WBCs. This is how the counter for WBC differential count looks like. The succeeding images show the different white blood cells that can be found in the patient's sample. In platelet estimate count, count platelets under oil immersion objective. The counted platelets in 10 consecutive fields accounts for the platelet estimate of the patient. Also, use the battlement pattern in counting the platelets. This image shows the platelets that can be found in the patient's sample. As shown in the image, this is the formula for the manual platelet estimate count. In using the Sysmex 330, click Manual. Enter new patient by clicking the sample ID and encode the last name and first name of the patient. Once done encoding the patient's name, click OK 2 times. Then mix the tube containing the patient's sample. Now uncap the tube and place it on the probe. Then click the aspirate button to allow the probe to sip the sample. Patiently wait for the probe to finish sipping before removing the tube from it. Once the machine has finished processing the sample, view results in the Explorer and search for the patient's name. Shown in the video is the results of the patient's sample generated by the machine. In performing prothrombin time, add 200 microliters of the white-capped reagent to the cuvette. Then click Enter. Press the arrow up button to incubate for 3 minutes. Now transfer the cuvette and place it on the reading cell. Click Enter and add 100 microliters of the plasma to the cuvette. Make sure that the tip will not touch the cuvette to avoid interferences. Now patiently wait for the result. Remove the cuvette and cover the reading cell. Afterwards, press Enter and click 0 to return to the main menu. In performing the activated partial thromboplastin time, add 100 microliters of the green cap reagent to the cuvette. Afterwards, add 100 microliters of the plasma to the cuvette containing 100 microliters of the green cap reagent. Then click Enter. Click the arrow up button to incubate for 3 minutes and click the right button to skip incubation. Now transfer the cuvette and place it in the reading cell. Then click Enter. Now add 100 microliters of the blue cap reagent or the calcium chloride. Make sure that the tip will not touch the cuvette to avoid interferences. Now patiently wait for the result. Once the result has been revealed, remove the cuvette and cover the reading cell. Remember to click Enter and click 0 to return to the main menu. For erythrocyte sedimentation rate, after collecting blood and inverting the black top tube 3 to 4 times, insert the ESR pipette to the tube and make sure the blood reaches the topmost portion of the ESR pipette. Once the ESR pipette is put in place into the tube, allow it to stand for 60 minutes. After the 60 minutes, read and interpret the results of the patient.